Hello and welcome. In this talk, I'm going to explain exactly what chemoproteomic drug discovery is and how our lab uses chemoproteomics in our drug discovery efforts. First, I thought it'd be good to start off with an introduction to what drugs actually are. Inside your cells, you have proteins, which are biomolecules that carry out almost all the essential tasks for life. In fact, 96% of all biomolecule drug targets are proteins. That's because most diseases result from abnormal protein function, either dysfunction or dysregulation. 89% of all FDA drugs target proteins. So drugs work by correcting the abnormal protein function or expression. A given cell has 10,000 different types of proteins, though. So this makes it incredibly difficult for us to understand which protein is targeted by a specific drug. To overcome this challenge, we use chemoproteomics, which enables identification and quantification of almost all the proteins inside a cell. So chemoproteomics is a variety of techniques. Today, we're going to focus on mass spectrometry, which allows us to detect drug-protein interactions. Mass spectrometry works by measuring the mass or weight and the abundance of all the proteins in a given sample. We then use the predicted mass for all the proteins from the human genome to then figure out the identities of their measured proteins from our sample. So it's an incredibly powerful technique that enables us to measure drug-protein interactions. As an example of a recent project in our lab, I'm going to tell you the story about Curcuzone D. So Curcuzone D is a molecule that previously exhibited anti-cancer properties, this molecule is isolated from a natural source, the Barbados nut, which is used in traditional folk medicines. Nature is in fact the best source for new drugs, since 50% of all FDA drugs were developed from molecules isolated from natural sources. So our question was, how does Curcuzone D kill cancer cells? In other words, what are the key proteins that Curcuzone D targets to kill cancer cells? Our chemoproteomic experiments using mass spectrometry revealed that BRAT1 was a key protein target of Curcuzone D. BRAT1 disproportionately helps cancer cells grow and survive by repairing their DNA. In fact, previous studies have showed that BRAT1 plays key roles in cancer progression, proliferation, metastasis, and patient survivability. So this is the plot from our mass spectrometry experiment. Each dot represents a different protein. So this identification experiment identified BRAT1 as a key target of Curcuzone D inside the cancer cell. We then did a fluorescent microscopy experiment. So the green indicates DNA damage. So we treated the cancer cells with etoposide and or Curcuzone D. Etoposide is an approved drug that causes DNA damage. So we see in the etoposide only experiment that there's some DNA damage. When we co-treat with both Curcuzone D and etoposide, there's a huge increase in DNA damage because the cancer cells can no longer repair the damage to their DNA, and then they subsequently will die. So this is an exciting study for us because Curcuzone D is the first known drug molecule that inhibits BRAT1, this promising anti-cancer drug target. So this is a new therapeutic strategy that we can hopefully use to develop better chemotherapies. There's a lot more work to be done to optimize Curcuzone D to be a successful drug, but we're excited about our preliminary studies. So if you're interested, you can check out our recently published paper in the Journal of the American Chemical Society. Finally, I'd like to acknowledge everyone in our lab. As a lot of people contribute to the science, including our collaborators at Purdue. If you're interested in learning more, feel free to email me at bdwyer@scripps.edu, follow us on Twitter, or Google us and check out our website. Thanks for listening.